12th Science, Chapter 14, Biomolecules, Part 2, Proteins and Nucleic Acids.
Hello students, I am Madhusena, your chemistry teacher. In this video, we will discuss about few more biomolecules, proteins and nucleic acids.
Proteins are a class of biologically important compounds. They are condensation polymers of about 20 different alpha amino acids, which are linked by peptide bonds. A peptide bond is formed when the amino group of one amino acid molecule interacts with the carboxyl group of another amino acid molecule. This reaction involves the elimination of a water molecule. Thus, the interaction between the amino group and the carboxyl group of two of the same or of different amino acids results in the formation of an amide linkage. That is, an NHC double bond O group called a peptide linkage. The product formed when two amino acids interact is called a dipeptide. The name dipeptide indicates a peptide bond that is formed between two amino acid molecules. For example, when glycine combines with alanine, a dipeptide, glycylalanine, is formed. Similarly, when two glycine molecules combine, glycylglycine is formed. Note that, by convention, the amino end is shown on the left and the carboxyl group is on the right of the resulting dipeptide. Also note that the peptide bond is planar. The CON and H atoms are all in the same plane along with the alpha carbons. Let's work on a practice problem. Show the formation of the peptide bond that forms between alanine and serine. Keep the amino acids in the order given here. It may help to start with the peptide backbone. Then look up the side chains and include them in the correct order. The alanine serine, represented as aliser dipeptide, results. Three amino acids linked by two peptide linkages are called tripeptides. The tripeptide, glutathione, protects the cells from oxidizing agents. It is formed from glutamic acid, cysteine, and glycine. Similarly, when 4, 5, or 6 amino acids are linked, the products obtained are called tetrapeptide, pentapeptide, and hexapeptide respectively. When more than 10 amino acids are linked, the product obtained is called a polypeptide. Thus, depending upon the number of amino acid residues present per molecule, peptides are classified into dipeptide, tripeptide, tetrapeptide, and so on, up to polypeptides. Polypeptide chains that have either more than 100 amino acid residues with a molecular mass greater than 10,000 or less than 100 amino acid residues with a well-defined conformation are referred to as proteins. Let us now look at the classification of proteins. On the basis of molecular shape, proteins are broadly classified into two classes: fibrous proteins and globular proteins. In fibrous proteins, The polypeptide chains are long and thread-like and run parallel to each other to form fibers. These parallel chains are held together by hydrogen bonds and disulfide linkages. Due to their strong intermolecular forces of attraction, fibrous proteins are generally insoluble in water. Important examples of fibrous proteins include keratin, found in hair, nails, skin, etc. Myosin, found in muscles. Collagen, found in tendons. And fibroin, found in silk. Thus, these proteins form the structural material of animal tissues. In globular proteins, the polypeptide chains coil around to give a roughly spherical shape. These are soluble in water due to the presence of hydrophilic groups. Globular proteins maintain and regulate life processes. For example, insulin regulates the blood sugar levels in the body. Hemoglobin and albumin are some other examples. Let us now discuss the three-dimensional structure of proteins. The structure of proteins is studied at 4 different levels, in the ascending order of their complexity, as primary, secondary, tertiary, and the quaternary structure. Let us first discuss the primary structure of proteins. The specific sequence of amino acids in the polypeptide chain reflects the primary structure of proteins. Here, we see the specific arrangement of amino acids of the enzyme lysozyme. Every protein has its own unique amino acid sequence. Any change in the sequence of amino acids creates an altogether different protein. The secondary structure of proteins refers to the arrangement of the polypeptide chains in space. One important type of secondary structure is the alpha helix, illustrated here in a ribbon diagram. Each polypeptide chain is coiled to form a spiral structure called alpha helix. The alpha helix is a right-handed helical chain in which the hydrogen bonding occurs between different pairs of the chain to hold the helix together. The hydrogen bonds occur between the oxygen atom of the carbonyl group of one amino acid residue and the hydrogen atom of the NH group of a different amino acid residue on the adjacent turn of the helix, as shown here. Another common secondary structure in proteins is referred to as beta-pleated sheet structure. The beta-pleated sheet is nowadays referred to as the beta structure. The arrows in the ribbon structure here show the beta structure. The contraction of the peptide chains results in a pleated sheet. The chains lie side by side and are held together by hydrogen bonds. Such a structure is called a beta structure or arrangement. The tertiary structure of proteins describes the further folding of the secondary structure. The tertiary arrangement of helices and sheets is held together by hydrogen bonding, disulfide linkages, van der Waals forces, and electrostatic forces of attraction. Shown here is the tertiary structure of proteins. The further folding of the secondary structure gives rise to two major molecular shapes, fibrous and globular proteins. Let us now look at the quaternary structure. Some proteins consist of multiple polypeptide chains called subunits. The quaternary structure describes how the subunits are arranged in space relative to one another in the complete protein. Hemoglobin is an important example. As shown in the illustration, the four protein chains of hemoglobin are arranged in a compact bundle. A protein found in a biological system with a unique structure and biological activity is referred to as a native protein. In some respects, proteins are rather fragile. Temperature or pH changes can cause a disruption of the hydrogen bonds. When hydrogen bonds break, the globules unfold, the helices unravel, and the beta sheets flatten out. This is called protein denaturation. When proteins denature, the primary structure, the specific amino acid sequence, remains intact. However, the secondary and tertiary structures come apart. Denatured proteins are unable to carry out their biological function. If you have ever boiled an egg, you might have witnessed protein denaturation as the egg white solidifies. Enzymes are proteins that catalyze reactions, speeding up the reaction rate. Enzymes speed up reaction rates by reducing the activation energy. Most enzymes are globular proteins. Nucleic acids are biomolecules present in all living cells and are responsible for transmitting genetic information from one generation to the next. In other words, they are the carriers of heredity. They are found to exist along with certain proteins. Proteins, along with the nucleic acids, constitute nucleoproteins. Now, in this context, let us look at the chemical composition and structure of nucleic acids. The basic components of nucleic acids include a pentose sugar, a phosphate group, and nitrogen-containing heterocyclic compounds called nitrogen bases. Depending on the type of the pentose sugar present, nucleic acids can be broadly classified into DNA deoxyribonucleic acid and RNA ribonucleic acid. Examine structures 1 and 2 given here. From the structures, we can say that in DNA, the sugar is beta-D2-deoxyribose, whereas in RNA, the sugar is beta-D-ribose. Let us now look at how these two nucleic acids differ with respect to nitrogen bases. There are 4 nitrogenous bases present in DNA: adenine, guanine, cytosine, and thymine. There are also 4 bases in RNA. In RNA, the base uracil is substituted for thymine. Note the structural similarities between adenine and guanine and between cytosine, thymine, and uracil. On the basis of the structural similarities among the nitrogen bases, they are broadly categorized into purines and pyrimidines. Adenine and guanine are purines, while cytosine, thymine, and uracil are pyrimidines. A nitrogenous base attached to the C1 prime carbon of the sugar is called a nucleoside or a base sugar unit. The nucleoside, adenosine, is shown here. Note that we number the sugar carbons in nucleosides as 1', 2', and so on as shown. A pentose sugar with the nitrogen base at the C1 prime carbon and a phosphate group at the C5 prime position is called a nucleotide, a base sugar phosphate unit. Look at the structure of a nucleotide of DNA and RNA. You can see that there is only one hydroxyl group attached to the sugar ring of DNA, while there are two hydroxyl groups attached to the sugar ring of RNA. Try to answer this question on your own before checking the answer. What products would be formed when a nucleotide from RNA, which contains cytosine, is hydrolyzed? Start with the correct nucleotide structure. Hydrolysis of this nucleotide would produce phosphoric acid, beta-D-ribose, and cytosine. Nucleotides are linked together by phosphodiester linkages. These nucleotides, which are linked by phosphate ester bonds, connect the 5' phosphate group to the hydroxyl group of the sugar at the 3' position, as shown in the diagram. The backbone of a polynucleotide chain is a repeating sequence of sugar phosphate. The sequence of nucleotides in the chain of a nucleic acid is the primary structure of the nucleic acid DNA or RNA. The sequence in which the 4 nitrogen bases are attached to the sugar phosphate backbone of a DNA strand is called the primary structure of DNA. DNA forms a secondary structure in the form of a double-stranded helix, like a twisted ladder. 2 strands of DNA interact to form the double helix, where the sugar phosphate backbone form the rails of the ladder, while pairs of nitrogenous bases, either purines or pyrimidines, form the rungs or steps of the ladder. James Watson and Francis Crick determined the double helix nature of DNA using X-ray crystallographic data collected by Rosalind Franklin. The 2 strands of DNA are held together by hydrogen bonds between specific pairs of bases. Adenine always pairs with thymine. Cytosine always pairs with guanine. As the 2 strands are held together by hydrogen bonds between specific pairs of bases, the strands are said to be complementary to each other. Notice that the complementary strands run in opposite directions. Helix, RNA is single-stranded. The strand may fold back on itself to form helical motifs, such as stem loops, or hairpin structures. There are three important types of RNA in living systems. Messenger RNA carries genetic information from DNA to ribosomes for the synthesis of proteins. Transfer RNA brings amino acids to the messenger RNA. Ribosomal RNA is the RNA component of the ribosome where proteins are synthesized in the cell. Nucleic acids play a number of important biological roles. They are the chemical basis of heredity, storing genetic information while also allowing for changes between generations. Each species has a unique genetic code with a specific number of chromosomes. DNA copies itself or self-replicates during cell division so that identical copies of the genetic code are transferred to the daughter cells. Nucleic acids are vital for protein synthesis. DNA stores the code for the amino acid sequence, while RNA molecules link the amino acids together. Each individual has a unique DNA sequence. Information regarding the sequence of bases on DNA is called DNA fingerprinting. Through DNA fingerprinting, it is possible to determine if two samples of DNA belong to the same person, two related individuals, or to unrelated people. DNA fingerprinting has important uses in identifying criminals, in determining paternity, identifying accident victims, and in the study of evolutionary changes. Based on the data shown, did any of the suspects leave DNA behind at the crime scene? Based on the DNA fingerprinting, we can eliminate suspects 1 and 3 as their patterns do not match the DNA found at the crime scene. It seems likely, however, that suspect 2 may have been at the crime scene.